Hello, a warm welcome to everyone who is joining our session today. My name's Helen Evans. So without further ado, I'm very pleased to welcome Connie to today's session. Connie's joining us from the States. Thank you very much for doing this on your Saturday morning. So for those of you who don't know Connie, Connie Lee is the founder and CEO of the Alliance to Cure Cavernous Malformation. And that's the US-based organisation working to inform, support and mobilise those affected by Cavernoma and to drive research for better treatments and a cure. As CEO, Connie has worked with the international research community, pharmaceutical companies, clinicians and global patient organisations to perform productive collaborations and begin clinical trials. And Connie will be speaking to us today on what is the latest research telling us about what medications can help. And just on a personal note, Connie's been working in this field for years. I first met Connie when I was first with CAK back in 2019. And it's just incredible, Connie, to see how much you've grown the organisation in recent years, just extraordinary. And the strides we're seeing being made and how you're pushing those forward. So it's we're incredibly appreciative of that here in the UK. So I'm now going to hand over to you and give you the floor.
Thank you, Helen. It's a pleasure to be here. I love talking to CAUK every time I have the opportunity and when I'm invited. And I want to share. Here we are. So yes, I have been running the what started as Angioma Alliance and is now the Alliance to Cure Cavernous Malformation for 21 years. It's certainly been a major chunk of my life, and certainly it's always been my passion since my daughter was diagnosed. She was diagnosed in right at the turn of the century on January 4th, 2000. And since then, there has been nothing in my world that's been more important than trying to advance care, community, and research for Cavernoma. So this topic that was assigned to me is, what is the latest research telling us about what medications help? And I moved a little bit beyond that as well. So I just want to give you an overview of what I'm going to be talking about today. I'm going to be talking about a little bit about what medicines might be able to do, and then give you an overview of what the clinical trial process is like. So once you have identified, as Doug mentioned, the medicines that might have efficacy in animal models, what happens after that? I will go one by one through the current pipeline of treatments that are in some form, shape, form, or nearly shape form of being tried in humans, and then also touch on some other treatments that are out there that people are thinking about that probably need a lot more work, but that could potentially be game-changing. And then I am going to talk about some ancillary things that need to happen. And Doug talked about nanoparticles a lot, things along those lines where the drugs that are being explored and investigated may actually have some utility with Cavernoma if there was an adjunct, if something was added to make either direct them directly to the lesion, or there are some other things that I'm going to mention as well. So what could a drug do? Or a treatment? It doesn't have to be a drug. What should a treatment do? We always think about, well, we need to stop hemorrhage. We do need to stop hemorrhage. We have other things, though, that medicines can do. And it could be that we have choices, that not every medicine will do the same thing. Not every treatment will do the same thing. And so we are going to probably stepwise move our way toward a cure, where some medicines will do a little bit of something, others will do a little bit of something else. Some medicines will do multiple things, but perhaps some things better than others. The idea here is, let's list out the choices. What is it that a medicine or a treatment might be able to do? So strengthening vessels and reducing the permeability, so in other words, the leakiness of the vessels, may be an important first step. And what we're hoping that type of treatment might do is reduce symptoms. And by symptoms, I'm talking about seizure and headaches, the things that come and go depending on the location of your lesion, keeping them quiet, keeping your condition more stable. The second thing, obviously, I've already mentioned is preventing hemorrhage. And for us, we understand that once someone has had a hemorrhage, their chances of having a second hemorrhage dramatically increase. In brainstem patients with brainstem lesions, that number is quite high. Over the course of 3 to 5 years, 1 in 3 people will have a second hemorrhage, and that is a very scary possibility. So we are looking at medicines that will prevent that second hemorrhage. So they are directed at folks who have already had a hemorrhage. We may have medicines that slow or eliminate the development of additional lesions. When people have the familial or hereditary form of CCM, invariably they develop more lesions over time. So by the time someone is in their 60s, they will have more lesions than when they were 10. And so we might be able to stop that process, but with some sort of treatment or medicine. There's another thing that we're looking at is slowing the growth of lesions. Doug mentioned this for different things, for gene therapy and for PIK3CA inhibitors. So stage 1 lesions are just tiny little dots on your MRI. Stage 2 lesions are the ones who have developed into a more mulberry-shaped lesion. And so we want to stop them when they're little dots, because at that point, they're not causing trouble. They're 1 millimeter, 2 millimeters at best. We want them to stay that size if they have to be there at all. And then, of course, one of the holy grails for us is to actually shrink lesions, make them smaller than they were. If they are mature lesions that are already looking like mulberries, can we make them smaller and perhaps even make them go away? Finally, there are 2 things that are also potential. One of them is to remove Hemocidrin. I'll go into that a little bit in more detail as I talk about one agent that's being looked at for that. And then the second is the thing you just heard a whole hour on, which is to fix the genetic mutations that cause the familial and the sporadic form of the disease, depending on whether it is in your entire body. The sporadic somatic form is just in the lesion itself. So what is a clinical trial? So as Doug mentioned, his job is to get things to the point where the animals are being effectively treated with a medicine or some sort of treatment. There's a lot of work that goes into that ahead of time. There's a lot of testing of these medicines in cell cultures that resemble the types of problems that we have with CCM. So human HUVEC cells is what they're called, and a lot of biology that happens to try to understand the mechanism by which a drug might be helpful. And then it goes into animal models. In CCM, the animal model progression is from worms, nematode worms, to fish, zebrafish, to mice. And that, by the time it gets to mice, it is probably already looking like a pretty good candidate, because mouse experiments, as Doug mentioned, are expensive. One doesn't start a mouse experiment until you have some really good thinking behind why it is that something might be helpful. So let's say your mouse experiment was amazing, and it is time to start discovering whether or not this has some applicability, works in some way in the human condition. First, you need to make sure that the substance that you're using, as Doug mentioned, is being manufactured in a way that's safe for humans. So that means following good manufacturing processes, having it tested to make sure that pill after pill after pill, if it's a pill, is identical. And then once you have that in hand, and that sometimes takes a while, the other part of this is it also needs to hold together as a pill, or you have to find a different way, maybe a gel cap. Perhaps it needs to be an injection because it will never hold together as a pill. There's so much that goes into making drugs. Once that happens, then it's time to start testing in people. Patients are not the first ones that wind up taking any medicine. When a medicine is developed brand new, the first people who will take it are healthy individuals who do not have the disease, and they take an escalating dose. That is to make sure that the side effects, and drugs almost all invariably have some sort of side effect, that the side effects are not horrific, or that they're not even significantly bad enough that it would prevent the medicine from being accepted by the population that it's intended for. Those trials are called phase 1 trials and typically involve somewhere between 20 and 80 patients and take just a few months, just enough time to find out, is this a safe drug? Then we start with our patients. Phase 2 trials. In this graph, it says 100 to 300 patients because our disease is rare. Frequently, phase 2 is divided into phase 2A and phase 2B. Phase 2A is when the primary thing that folks are looking for is it safe for our patients? Are the patients who are taking it, tolerating it? Is it not? Is it causing possibly even more hemorrhage? Is there something unexpected that's happening with this medicine in the patient population that means that it should not move forward? Phase 2A then looks a little bit at whether or not there's actual change in the disease process in this small population. As we move into phase 2B, then we move out into more in the 100 to 300 patient realm. Phase 2A, what we've been seeing is somewhere between 60 and 80 patients are in phase 2A before the trial can expand. And then in the phase 2B part of a clinical trial, we're looking to see, what is the drug doing? Is it making a difference? And there aren't enough people in a trial at that point in order for us to see for sure that the drug is working, but it's enough to let us know whether we should start looking for large groups of people to be taking the medicine. And that's where phase 3 starts, when the regulatory agencies say, all right, we're seeing that you've seen enough of a difference in your patients. They approve the beginning of a phase 3 trial, which then lasts another 1 to 4 years, depending on the medicine, that will then look at, all right, is this working? What's it doing? Is it better than anything else that's out there? Doing some, or at least different from anything else that's out there. And at the end of phase 3 is when finally a drug is considered for approval for public use. So that's when, at the end of phase 3, if a drug is approved, your physician can go ahead and write a prescription, and you will be able to take that to a pharmacy and get the prescription filled and get the medicine to you. There is another phase that comes after that, because it's important to continue following patients. And phase 4 is post-market, when the drug is out there and readily available to patients, just continuing to monitor to make sure that there is still not anything rare and unexpected that might be happening as the result of taking a medicine. Drugs have been pulled from public use after market because some unexpected side effect, for example, a heart side effect, has happened that warrants the drug being removed, because or warrants a label on the drug that's called a black box warning that lets folks know that there is the potential for a very rare, usually side effect, but a very serious one. Hopefully the drugs that are approved, the treatments approved for CCM, will not have to worry about that. Hopefully all of them will go on to be quite safe and not need black box warnings or not need to be pulled. But it's good to know that the investigation examination still continues even after people in the public are taking it. So what do we have going on? Look at that. Look at that. We have quite a bit going on already with our patients. And I am, again, going to step through one by one each of these so that you can have a sense of where we are. 5 years ago, this was pretty much, there's a number of things on there, but there was nothing in phase 1 even. It was all preclinical. So the mice were enjoying their medicine, but humans were not yet. We have our potential treatments divided into different categories depending on what it is that they are intended to do. So the first category right now is the most popular, It is a Rho kinase inhibitor. Rho kinase inhibitors are re-regulate a chemical balance. Like, for example, when the CCM1 gene is not functioning properly, Rho kinase is a different protein that is in the system, in the endothelial cells in greater quantities than it should be. By inhibiting it, you're restoring an equilibrium that the hope is that it will, for atorvastatin, what they're looking at is it will prevent the second hemorrhage. For NRL 1049 and GV101, they're looking even further, that perhaps it will stabilize the lesions or even shrink the size of the lesions is the eventual hope. They are very far from doing either of those things yet. So atorvastatin is a statin drug that's used for cholesterol, and has been shown in mice to be potentially beneficial for humans. It has been in trials for a while now. I think the trial started in 2019 at the University of Chicago only. It's a phase 2A trial, so mostly a safety trial, but also looking at the potential for there being an effect. And at the same time, this trial is trying to see whether or not a special type of MRI sequence called QSM would be an appropriate way to measure the change that happens in a clinical trial. Because as we all know, hemorrhages are far apart. A clinical trial typically can't wait to see if enough people hemorrhage. It would either need to be really, really big, or it would need to go on for many, many years. And so we're looking at what are called surrogates, biomarkers, to determine whether or not there's been a change. QSM will measure the amount of iron that's in the brain. Doug showed you such a thing with mouse brains that's not QSM. They use a different way of doing it with a staining process. With QSM, there is no staining. There isn't even contrast agent used. It's just a different way of manipulating the MRI software so that it is focusing in on the iron that's in the lesion and seeing if that changes in and outside of the lesion. And if that remains if the amount of iron remains the same, that means the lesion is stable. If it increases, that means there has been activity in the lesion and perhaps even hemorrhage. So the atorvastatin trial, does atorvastatin make a difference? Is it safe for our patients? And does QSM have some clinical applicability? Does that level of deposition of iron deposit correlate with changes in clinical symptoms in the patients? The next two, NRL 1049 and GV101, are medicines that have never been used in people before. They are both medicines that are being developed specifically for Cavernoma. NRL 1049 is a medicine that used to be called BA 1049. It was developed preclinically by a company called Biomarkers, who we brought in to the CCM field. They were developing a Rho kinase inhibitor in their lab, and we said, hey, we're here. Why don't you look at our indication to see if it works for us? And they were enchanted. And we were able to connect them with our mouse labs, our academics, including Doug. And in fact, it was primarily Doug, in order to test their then BA 1049 in the mice. Again, very promising results. Now that they're moving into human trials, that isn't the domain of a company like Biomarkers. It's now being licensed to a company called Neurelis. And Neurelis is taking this medicine and moving it forward into trials. It is already being tested in healthy adults. And again, I think for the most part, all of the treatments that you're going to hear about that are pills are, with the exception of propranolol, but even with propranolol, are only at this point being tested in adults. It is very, very difficult to get approval to test in children because of their vulnerability. So first we would get a drug approved for an adult patient, and then we would move forward with moving that medicine and moving it forward for children. So NRL 1049, been around for a long time that we've known about, being developed specifically for CCM and in phase 1 trials. Then just about 6 weeks ago, a second company came out and made an announcement that they have a Rock2 inhibitor that is a Rho kinase 2 inhibitor. So it's a more specific inhibitor of Rho kinase that is in phase 1 trials already also, and that they would like to target CCM. And in fact, when they made the announcement, their idea was, let's target brainstem CCM. Again, as I mentioned, the rehemorrhage risk for brainstem CCM is much higher than even for lesions that have hemorrhaged outside of above the brainstem. We have been working with them since their announcement. They announced in the morning, they emailed us in the afternoon and said, help us. So we are now working with this company as well. It is a partnership between two companies, Graviton and Ovid, to move their medicine forward. There's a lot of work that needs to happen. It's being tested in humans that are healthy, but there's going to be a lot that needs to happen before it's tested in humans with CCM, probably more so than with the NRL 1049. Those two compounds work in the same pathway, but they are different enough that they warrant having their own clinical trial. So that's the ROCK inhibitors already being tested in people, all of them. We have this other trial, REC-994, which is being run by a company named Recursion. This is one that we are particularly proud of because we've known the CEO of Recursion since he was a graduate student presenting his papers at our scientific meetings. And then he went on to found this company, and this company now has many drugs in their pipeline. I believe 4 of them are already in clinical trials. One of them, the first one, is for CCM, and it's called REC-994. This is a drug that is intended to reduce symptoms. So it's stabilizing vessels. It is hopefully reducing the leakiness of the vessels. It is not intended to be taken in those first 3 months after a hemorrhage and make some big, huge acute change. This is intended for long-term use to keep things quiet. It is an antioxidant. It's like vitamin D on steroids, is what I continue to describe it as. And just June 13th, so just like 11 days ago, we finished the enrollment of their phase 2A trial. So we have 15 sites around the United States where patients are being enrolled. This is a 1-year trial. People need to be on the medicine for 1 year. And then at the end of the year, they're allowed to stay on through a trial extension. But really at the end of 1 year, when everybody's done with that, they will be looking to see whether or not the drug is safe and if it shows any signs of efficacy. And at that point, the trial will widen out to, if it is safe and effective, to more patients again. The reason that it was fully enrolled is because our patients stepped up in a big way. They finished their enrollment ahead of schedule. And this is encouraging for us in many ways. We know our patients will show up. Other industry partners like Neurelis and Ovid and Graviton are seeing that our patients are stepping up and participating in trials, which makes them much more likely to want to stay in our field, continue work on our disease, and makes other companies looking in from the outside say, hey, this is a group who's got their act together. We want to make sure that we're in on that too. And if we're developing something that looks like it may have efficacy for this disease, we're not going to pick disease B or C. We're going to pick Cavernoma as our choice for the first thing to try it on. So that's REC-994, particularly proud. If you look on our YouTube channel, there are a number of presentations already on there from people from company employees, some of which are no longer there, some of which are on the compound itself. And then Chris Gibson, the CEO, is going to be the keynote speaker at our upcoming scientific meeting, and will be That will be videoed and on our YouTube channel probably 2 or 3 weeks, but somewhere around the middle of July, middle to end of July. All right, next, low-dose rapamycin and alpelisib. Doug did a great job, and I'm so thankful, of talking about PIK3CA inhibition and why that's important. That rapamycin that he mentioned works a little bit further along in the pathway beyond PIK3CA in a pathway called mTOR. Rapamycin is what is the drug that's being tested in the mice in his lab in order to see whether or not it has an effect on CCM, mice with CCM lesions. As far as I know, that work is wrapping up. So the mice are pretty much done taking it, and now it's time to look at the mouse brains and figure out whether or not there's been a change. At the same time, the belief is that this is really we're going to see changes as a result of rapamycin because it's been used so widely in other diseases that are similar, overgrowth syndromes, where people have overgrowth of lymphangiomas or AVMs, arteriovenous malformations, which are similar enough. So the trial is seeking funding in the US to see if we can get some money out of our National Institute of Health. Rapamycin is a generic drug. It is not owned by it's not under patent with any particular drug company. So the money that comes that has to go into a clinical trial has to come from government. That funding there's far less of it. There's less motivation to give it out. So it may take a while before we can get rapamycin into patients. The Alliance to Cure is looking at perhaps contributing a little toward a pilot study. We'll see how that turns out once the mouse studies are finished. Alpelisib is a specific PIK3CA inhibitor that is under patent and owned by Novartis. It has not yet been tried, to the best of my knowledge, in any CCM mouse model. That is something that we're looking at potentially forwarding if we can get the go-ahead from Novartis. Companies frequently don't like to have their drugs tested for a new indication while they are still under patent for something else, in case something goes wrong. They don't want their drug to be pulled from the market as the result of a side effect that's discovered in a different disease. So it's a lot of negotiation, and sometimes it has to happen behind the scenes, and sometimes the drug companies themselves are doing the work. So that's a story to be continued. It is phase 1, and the reason I put both of those in phase 1 is because they have been already tested in healthy volunteers. So now we're looking to see whether they can be tested in our patients. CD52 is a microRNA-27 target blocker. This is a medicine that is actually a subcutaneous injection, but it wouldn't be something given every day. The injection is very similar to what you would have for insulin, or now those very trendy weight loss diabetes drugs. At least they're trendy in the United States. So subcutaneous injection is not sticking something in your vein. It's actually sticking something into the fat tissue that most of us are carrying around our waist or in our thighs or somewhere else. And it had some striking results in mouse models where the mice were still quite young, and in terms of delaying the development of more lesions, and even giving some indication that it could shrink lesions. So right now, the medicine the Alliance to Cure is funding a test of this medicine in mice that are older. So as Doug mentioned, mice that already have developed lesions that are stage 2 lesions, can it make a difference in those? Or is it only something that can apply to the very young mice when they're in a phase of rapid growth of blood vessels? An angiogenic phase is what that's called. Or can it actually help with mature lesions? And that's a story where we will have an answer probably by the end of the year, one way or the other. If the story looks good. If it looks like it is making a difference in these mice, then we will have to expand the mouse trial for a little bit more in order to get more mice involved and check out other effects on other organs, et cetera, et cetera. There's a lot that would still go into it before we start testing in humans. Propranolol is a trial that has completed its phase 2A. It's a little into 2B as well. The trial itself was contained to a number of different sites in Italy. And it appeared to show that propranolol can make a difference, particularly in the development of new lesions. So that trial currently is seeking funding to expand. They could expand directly into phase 3 from where they are right now. So into a much larger trial. They're seeking funding in the EU. They're seeking funding in the UK. After the trial was started, that's when the mouse studies came to be. It's the reverse of how it usually happens, but that's because propranolol had been prescribed off-label for a few patients and seemed to make a difference. And it's already safe and approved and on the market for blood pressure control and for tremor and for a bunch of other things. So trying it in humans wasn't a huge risk step. We're hoping to get some additional funding to get it off the ground. So far, the grants have not been successful, but that doesn't mean they won't keep trying. Focused ultrasound is another very exciting potential treatment that is not a drug. It is very similar in administration to what gamma knife is, to what stereotactic radiosurgery is, in that you get into a tube that looks kind of like an MRI machine, and there are focused beams going at the lesion itself. The difference with focused ultrasound is that there's no radiation involved. It really is just ultrasound. So ultrasound is truly sound. It's vibration. There are two ways, and please go back again on our YouTube channel. There is a graduate student at the University of Virginia in the US. Her name is Delaney Fisher. She's amazing. She did a great presentation at our patient conference last November about the different ways that focused ultrasound could potentially help cavernous malformation lesions. A sonosensitizer could be put into a person's body, and that means that it's a substance that is more sensitive to sound waves. And so when it's in the body, if you can direct the focused ultrasound toward the lesion, if there's sonosensitizer in there, that sonosensitizer may be able to release substances, and the substances could be things that actually destroy the lesions. So focused ultrasound may, again, as a gamma knife, but more safely, be able to destroy lesions without surgery. And for people who have sporadic CCM, sporadic cavernoma, just one lesion, this could be curative. The same, a different mechanism is microbubbles. Microbubbles are truly just bubbles that have gas in them. When the focused ultrasound is focused on them, they expand and contract, opening the blood-brain barrier. And once the blood-brain barrier is opened, it allows for more easy access of medicines that could potentially otherwise be toxic. For example, the PIK3CA inhibitors are cancer drugs. These are not mild drugs like, over the counter drugs that you might take. These are drugs that are known to have serious side effects. But if you can contain them to just the lesion, then that becomes potentially some sort of treatment. And then finally, low dose aspirin is being explored as a treatment. That is counterintuitive to those of us who, for the longest time, have said, stay off blood thinners because they're going to make you hemorrhage. The thinking now is slowly coming to the place where there's indications that clotting in the lesions, in these slow flowing blood vascular lesions, the clotting may actually be a precipitant of hemorrhage, and that if you can slow down, prevent clotting, that you may get some effect on hemorrhage rates. And in fact, it looks as though people who are on antithrombotics, so medicines that prevent clotting, have no greater risk of hemorrhage, apparently, and they may indeed have a lower risk of hemorrhage. So in the UK, there is hope that some funding can come for that trial specifically. And that, as far as I know, is the one place where we're looking to get funding for a low-dose aspirin trial. So additional preclinical trials. These are out there. These are not yet ready. These need to be tested in mice. Have not had any mouse trials yet. Metoprolol is a medicine that's like propranolol. It's another beta blocker used for blood pressure. The thinking behind metoprolol is that it may be effective at a lower dose than propranolol. Propranolol is not all that well tolerated by our patients who don't have high blood pressure. The doses that we think are needed in order to make a difference with cavernous malformation lesions frequently cause folks to have blood pressure that's too low, honestly. And so metoprolol, we think, has the potential to have the same effect on cavernous malformation lesions, but at a lower dose. So that's something that we need to test in mice. Another one is Avastin. This is a stronger medicine that is already being tested in humans for a sister disease of ours called HHT, or hereditary hemorrhagic telangiectasia. It is intended to stop the growth of blood vessels, and testing it in mice will help us to see whether it can stop the growth. And it's one of those drugs where it has to regress lesions or it's not worth following up, because it can be, it is on the strong side of the drugs that are being tested. Let me see if I can move my slide forward. It just got stuck on me. All right. And now, I mentioned in one of my early slides, removing hemocidrin. So hemocidrin is that iron deposit that's outside of the lesions that have had leakiness or hemorrhage. We believe that hemocidrin is one of the precipitating causes for seizure in people who have cavernoma. We also think it may be one of the things that contributes to central pain syndrome in people who have lesions in the midbrain, in the thalamus, and in people who have spinal lesions. So if we can remove hemocidrin, we may be able to make a difference even post-surgically, even after people no longer have a lesion. And if we can remove it without removing extra brain tissue, which is the key, we might be able to help folks who have still post-surgical epilepsy get rid of that epilepsy. We may be able to help people who have pain from thalamic lesions or from spinal lesions get rid of that pain. Even with those lesions, we don't necessarily want to, those last two, want to remove them. Frequently removing them can be very destructive, but even having some relief from the pain would be life-changing for many of the patients that have these. So there is a medicine, let me go back one sec. There is a medicine that's being developed for other indications that can cross the blood-brain barrier and remove iron deposits. We are in talks with the company right now who are developing that to see whether or not we can test that in our mice. Along the same lines as focused ultrasound, and again, this is like something that we kind of recognized in the last few months, so it's very, very early, are tumor treating fields. This is electric fields rather than ultrasound fields that can disrupt cell division. We think, just within our alliance, that it has some potential for people with spinal lesions. There's a device that you need to wear. It's a little sticker thing. You can see it in the lower left corner that generates electrical pulses that would be tuned to the specific frequency needed to disrupt the cells in a cavernoma lesion, and not any other cells. So it would they get tuned to the specific pathology. So the endothelial cells that have lost tight junctions and have the mutations have their own vibration or their own electrical frequency. The generator would be tuned to that. You would wear the patch for as long as it takes to either we're hoping for as long as it takes to remove the lesion. That's the goal. It is currently being tested, and multiple cancers are being treated with this, and it is approved already for glioblastoma multiforme in the EU and Japan. I want to also step back to focused ultrasound. There's something I forgot to say. Focused ultrasound, there are 20 different facilities in the UK that have focused ultrasound machines. 4 of them are already using focused ultrasound in an approved way, regulatory approved, out on the market kind of way, for brain indications. And the brain indication is essential tremor. So essential tremor is being treated with focused ultrasound in the UK, in 2 facilities in London, 1 in Liverpool, 1 in Dundee, I think. Don't quote me on the last one, but it's definitely in Scotland. So it's not that it would be inaccessible to UK patients. There are places that could do it should it ever be approved. Tumor treating feels, you wear it, so you don't need to go somewhere special to get it. As I mentioned, there are other tools that we need to continue developing in order to make this work go faster, and also to have other treatment options that are safe, that can be safe. One of the things that's being developed are ways beyond mice. Mice are not human, and so very frequently things that work in mice don't necessarily translate to working in humans. And in fact, the statistics are actually pretty bad. It's like 90% of things that work in mice do not translate to humans. So I don't want to discourage anyone, but we can develop more tools that resemble the human condition. Right now, we're looking at something called an organoid, which is basically the replication of a cavernous malformation in the human form. It uses human stem cells, pluripotent stem cells, to create a cavernous malformation that can then go on to be used for screening treatments and testing treatments, mostly for screening, though, to see whether or not a treatment has some potential in the human form. I'm going to keep moving on because I see my time is getting short. We are also looking at biomarkers that will help us figure out who has CCM and definitely who might be the best candidate for a trial. So predictive biomarkers would allow us to see who is most at risk for hemorrhage. And those folks might be the ones who want to be enrolled in a trial or take a medicine after it's approved. This work has been going on for a good number of years in the US, and also now that it was part of the propranolol trial. And there is talk of getting even more work going in the EU to try to figure out what the best way is to measure through blood mostly. And in Boston Children's, they're looking at urine, whether or not somebody is at risk for a hemorrhage in the upcoming months. As Doug mentioned, there is efforts to develop different ways to deliver medicines. And I mentioned focused ultrasound. Nanoparticles can be coupled with focused ultrasound, or can work all by themselves if we can figure out how to deliver drugs to a specific location instead of having it have to go through the whole body. This is something that the Alliance to Cure is funding. We're putting money into the University of Pennsylvania in order to have them develop nanoparticles that can specifically target cavernous malformation lesions. Now, we're still at the point where we're not sure we can target brain vasculature. So it's going to be a ways before we move from, yes, we can target brain vessels, blood vessels, to, yes, within the brain, we can target cavernous malformation vessels. But we're starting this. And this becomes important when we talk about PIK3CA inhibition and some of the other medicines that have more toxicity. We want them just to work in the lesions and not to have any effects other places in the body. And then I think this is my next to last slide. We have our science meeting coming up in a week and a half. And it brings together researchers from around the world who will this year be discussing some of the things that I just mentioned. Gene therapy, disease models other than animals, but they're also going to be discussing patient and clinician priorities and what how we can advance and make sure we focus on the things that are of the most importance to patients. As you've heard from my presentations, there are more and more pathways that treatment exploration is going. But in the end, what we want is a convergence on a cure. We want all of these pathways to continue to see what's the most beneficial, but we don't want them to start straying off in places where patients would never do that. For example, gene therapy. Would a patient ever allow their child to have gene therapy? We don't know that, and that's what we're trying to assess. So that's a discussion we'll have at our science meeting, and we're looking forward to hearing about some very new things too. And Chris Gibson from Recursion is going to be giving our keynote speech, which will be on video, and you'll be able to watch it on YouTube around the end of July. So summary, explosion of treatment options that are being tested, developed. We have additional treatments that we want to test in preclinical settings in animal models or in organoids. We are developing organoids, biomarkers, targeted delivery systems that we hope will speed research and allow us to have more and more treatment options. And as you saw, the long list of the different things that a medicine might or treatment might be able to do. And then our science meeting in a week and a half, we're going to pick up where we left off in November and keep talking about these things. So I want to thank you all. This is always a pleasure, and I always get to talk long because there is so much to say. I'm going to stop sharing now.
Connie, thank you so much. It's been said in the chat, but it is so hopeful for our members to see, all these opportunities in development, and just even in the last few years, the extent to which things have come on. And I also just to say thank you for your role there. You talked about a scientist who'd first attended some of your conferences. I think he was a PhD student, and then to see now he's at the forefront of a new treatment is incredible. And I think it emphasizes how important patient advocacy groups are, because he wouldn't have been nurtured and supported to end up where he is without what you and your alliance have done. So it really shows that patient power. And as Connie said there, please, do have a look mid-July. Look out for the sessions from the conferences. There's going to be so much more to learn from. So looking at the Q&A and the chat, I can see we've got 6 questions so far, Connie. So I'll take you through them. So the first question, Connie, this might not be one for you actually, perhaps myself and for David, which is aspirin, statins and propranolol, although awaiting UK funding, can these be prescribed off-label, keeping in view they're being tested in phase 2 trials? David, I was just wondering if you are available to give your view on that as your advisor to us. I put David on the spot there, so he might not be.
If he can, I can respond to that.
Go on, Connie, you respond then. There we go. Thank you, David.
You can put me on the spot, but I don't know the answer to it, I'm afraid. I doubt they will be prescribed. Can they be prescribed off-label by doctors in order to treat Cavernoma? I'd very much doubt that doctors would feel able to do that.
I disagree, David.
All right.
David, and I want to say that propranolol is being prescribed around the EU, and I believe also in the UK, primarily to children who have Cavernoma. And the reason is that it is the only drug under consideration that is approved for use in children for anything. And so the doctors are feeling like, well, it's bad, it's kids. What do we got to lose? I think if Dr. Al-Shahi Salman were on the call right now, he will express his horror at doctors who would do that, because it does eliminate the possibility of the patients being in clinical trials and of us seeing whether or not a drug actually is effective. So it's only in a randomized clinical trial in that setting that we can understand the effect. The other thing that we have with statins is the dosage that they're testing is very high. And again, as I mentioned with propranolol too, the doses are very high. They're very different from what people get when they just have mild high cholesterol or a little bit of a blood pressure issue. So if you're using the lower doses, there's no effect of those two in particular. There is no effect. There's not intended to be any effect on the lesions. So don't. Don't.
I mean, I hope you're right.
And I think just in answer to that, as we've said earlier on, today's sessions, all our speakers are sharing their views. And I would say ask your clinicians. And anecdotally, certainly from some of the focus groups we've been doing recently, we do know, that there is prescription taking place. So, I think it'd be one to discuss with your clinicians. And Connie, David, really helpful to have both your perspectives there. And I think the thing we really need is just to get this all these trials completed, don't we, Connie? And then there's none of that ambiguity, and we can move forward definitively with those. So moving on to the next question. So can the antipsychotic olanzapine help with CCMs, especially with headaches? I'm guessing it would not be used if it is helpful because of the serious side effects. So Connie, is this a drug you're familiar with? And you could...
This is a drug that has never been mentioned in relation to CCM, and I really can't comment on whether or not it has any potential. I'm not familiar enough, and I'm also not a physician, so or a researcher. Honestly,
Yes, Nicola, thank you for that. Thank you for that question. It's one we can, as Alliance, have a look further into and see if we have information, but to the best of our knowledge, we're not aware. So next one from Marie. A wonderful presentation. I'd echo that. Yeah, it's been excellent and great to hear about the ongoing trials. Just a quick query on vitamin D in CCM. How does this help patients with cavernomas? Do you just give to patients who have a cavernoma already, or if familial CCM but scan and the scan's normal, so it's would you still take it? And are there any other vitamins that you would advise? I know Connie can't advise here, but you can share your knowledge. Are there any other vitamins that patients could take?
Yeah, so vitamin D is important. And the reason that it works is because it is an anti-inflammatory, it's an antioxidant, anti-inflammatory. And we like to keep inflammation down. This is the other area that I didn't have time to discuss, is the gut microbiome and making sure that you're eating healthy foods and not a lot of pre-processed, overly processed foods that have preservatives. All of that is an attempt to keep inflammation at bay, because we believe that, and I think it's been demonstrated, that inflammation can trigger hemorrhage. And so for vitamin D, get your vitamin D level checked. If your physician will allow you to have a blood test to see what your level is, if it's low, supplement. And yes, most people with CCM here in the States, they just take vitamin D. And I think that it has helped us to keep the hemorrhage rates down over the course of the 5 years or so that we've been suggesting folks do this. And there is no other vitamin that I'm aware of.
Excellent. Thank you, Connie. So the next question, how does propranolol help in reducing the risk of bleeding from cavernomas? And it says cavernoma, it says exactly cavernomas anomaly in normosensitive patients.
Yeah, and this is, again, a little beyond my expertise. This is Petra Magnusson from Sweden should be the one addressing this. It has to do with beta adrenergic pathways and lots of sciencey things that I don't understand. And again, we weren't necessarily saying that it addresses hemorrhage as much as it addresses the development of new lesions. It may stabilize lesions. So there's a lot more research that needs to happen with propranolol before it's really understood. As I said, the mouse work didn't start until after the patients started taking the drug. So it seems to be working in the mice too. And if you have an opportunity to talk to Petra or to someone else who is a scientist about it, then you'd probably get a better answer. Well,
Connie, as it happens, we've got David and I have a meeting with her in the diary actually for a few weeks' time. So it's a timely question.
Good. And David may even be able to answer this question already. I am not.
David, is there anything else you'd like to add to that answer?
I don't think I can offer anything to it. If I could just come back to the difference between Connie and me on the previous one, I think there's a difference in the UK from my own experience between clinicians in hospitals and GPs. And I think GPs are less likely to go against NICE recommendations than clinicians in hospitals are.
Mm. Maybe, and that's a really important distinction, is it? Because we know many people will be discharged to their GP. Although we see more children under the care of a neurologist, unfortunately, the caseload of adult neurologists can be very high, and same for the neurosurgeons, which can make it sometimes challenging for our members to get appointments. Thank you, David. So I'm going to move to a question in the chat now, and that's from Mark, our outgoing chair. Connie, thank you for your excellent presentation. I'm deeply encouraged by the range of treatments being investigated that you've outlined to us. My concern is that because asymptomatic cavernoma is so rare, and consequently the market for treatment so small, does this mean there is little incentive for pharmaceutical, medical companies, even state-funded bodies to fund research? Or have you been pleased by the willingness of such bodies to fund research? And then just a follow-up question, or is the range of research into treatment options a result of your charity's work?
It's a very complicated question, the economics of it, honestly. I'll share the anecdote of the latest group, that Ovid Graviton group, that are coming in with a new ROCK inhibitor. Their perspective is that CCM will be the first indication for this drug. It will be their way into the market, but that there will be numerous other indications after this. And so economically, then it makes sense for them to start somewhere where they think they can see an effect pretty quickly. So that's good. The second thing is that I don't know what pricing for any of the medicines that are being newly developed will be. So I don't know what the recursion drug will be. I don't know what the Neurelis drug will be, and I don't know what the Graviton drug will be. Certainly, they will need to gauge the price, or set the price of the medicine in some sort of balance with the effect that it has. So if it's curative, like gene therapy, you only have to give it once, the sky's the limit. But if it's something you have to take every day for the rest of your life, then it needs to be somewhere within reason in order for in our system for insurance to cover it, in your system for the NHS to pay for it. So there's that. If it's done daily, this is a more common disease on the side. As rare diseases go, we are on the more common side. So we're not like 3 people in the world. We are, several million people in the world. And then finally, there's a lot of again, the patient organizations drive this research. Engaged patients are what make this happen. Once the industry and academia and government agencies see that there is the ability to mobilize patients and get them involved in whatever form, whether it's clinical trials or other types of research, then there's more motivation to move it forward. Then there's the potential for some return on investment.
And Connie, just to say, at our AGM, Mark talked about our shift in focus to give greater priority to research. And that will be a really important part of our work, where we see pharmaceutical companies looking to bring their drugs to European markets. We want to be having conversations with them and making sure that patient voice is really heard as the regulator decides whether or not to approve the drug, and then whether they want to buy it. So I'd echo your point about the importance of the patient community here. So Connie, there's 3 more questions. I'm conscious we're just coming up to 3, but might you have time for just those last few questions?
Yes, I do.
Thank you. Our next session is at 4, so
it's only 10 a.m. in the morning here.
Bright and breezy, we appreciate it. Now, the next one, I'm just going to give an initial answer to, because it's there's also a point here that's specific to CAUK. So Ian, and just acknowledge Ian was our founder. So Ian, it's always fabulous to have a question from you. And he says, thank you for your talk, Connie. Would you say it is of importance to answer questions of new members diagnosed with Cavernoma so that a membership base can be built for research? So Ian, just on this one, as you know, we established, because you did, establish CAUK as a member-led organisation. I think for me, coming into the CEO role, what I've reflected on is the questions we ask of people to become a member of a charity, and that gives them voting rights, gives us information to enable us to contact and support them. And then there's the questions that we ask of people for a patient registry. And I think one of the things we need to think about as a charity, as we prioritise research more, what might a patient registry look like? Because I think at the moment we've asked questions as we would of our members joining a charity and a few more, but we've not got the questions needed for patient registry. We probably need to separate out the two and really think about defining what a really good patient registry would look like and be of value to researchers, both clinical and basic science researchers. So Ian, that's just my personal perspective in terms of my role at CAUK. Connie, do you want to give your thoughts as well?
No, I think certainly there is, and patients need to understand their disease. They do need to be motivated to participate. The Alliance to Cures website has as extensive information as one can find anywhere. And so, it would make sense to have folks be channeled in that direction in order to catch up on reading. The other thing is money. There is a true need for funding of a patient organization. You have seen what a difference it's made. The Alliance to Cure has functioned with a budget that was, it's significant, was $200,000 for many years. And then all of a sudden we hit some milestones that increase that, and our budget is now around $1 million a year. And the difference in what we can do between $200,000 and $1 million is just astronomical. I have a staff of 9 people. Not all of them are full-time, there's half and half, but our bandwidth to be able to make these differences can only happen if folks are generous. And we don't have government funding at all. Our money comes from private foundations, but a lot of over 55% of our funding comes from individuals who are giving gifts of $5,000 or more, so of major donors. And another 20% comes from people who are giving, $5, $10, $20. Money talks. So anything you can do to financially support CAUK, whether it's a bake sale or writing a check that is of significant size, is important. That is a big part of what's going to make a difference.
Connie, thank you for saying that, and hats off to you for how you've grown your organisation. That's extraordinary, the journey you've taken. And yes, as you say, funding is key. It's something we really are going to prioritise and see. We have taken a dip, and we're very, very keen to build our income back up, because we know what we can do with that. And your charity is a perfect example with when the money comes, but that was a long, hard slog. 20 plus.
It's been 20 years. I mean, it's 20 years to build up to this point.
And you deserve to be where you are. It's fantastic. So, Connie, we've got there's 3 more questions. I've had one offline, so I'll move on. So this one is a question about the scientific meeting, and just asking whether there be a summary of the main conclusions made that will be shared, or would you signpost people to the videos?
There will not, and there never is, and there are very few videos. The reason for this is because in our science meetings, people are really only allowed to share work that has not yet been published and peer reviewed. It's confidential. They're not allowed to take pictures even of slides. There's no recording, in order to allow for the most cutting edge work to be discussed. So this is all preliminary, and we can't scoop people, because then they won't get the publications and they won't get to keep their jobs and get their funding. So what the best we can do is publish the agenda. And when you see the titles in the agenda, you can have a sense of what the topics were that were covered.
Thank you, Connie. And just if there's been any confusion, there's 2 meetings that's going to be taking place. One is for patients and the other's for the scientists. So yes. The first, more information is available, but as Connie said, for very important reasons, the second won't be. So
let me just I should have expanded in the patient conference. Some of the very same presentations that are giving at this, presenters at least from the science meeting, will be presenting at the patient conference. So those will be on video and available.
Fantastic, Connie. So, Connie, you're very popular. There's a lot of questions. Are you okay for me to give you 4 more? Are you sure? Thank you. So the next one is, this is drawing on your knowledge of the research, and we're mindful you're not a clinician. Can propranolol be used for incidental cavernomas, and will the trial include incidental patients?
Propranolol has been used for incidental patients in an off-label way. The trial in the EU only included familial patients, and that is very specific to the EU because only familial CCM is considered a rare disease. So the funding that comes for trials from government sources can only be used in this, or at least in the way that this was set up, could only be used for familial disease. I don't know what they're going to try to do going forward, whether they can make sporadic CCM be part of the next trial, unfortunately.
Thank you, Connie. So this is one that's coming offline. Is there a medication that can help to counter increased occurrences of déjà vu and visual auras other than propranolol?
Those are seizure signs. So one would want to talk with your neurologist about what options there are in seizure. That's temporal lobe seizure symptom.
Excellent. Thank you, Connie. So next one. Do you know anything about drug treatment for migraine symptoms? It was mentioned on some webinar that we have CCMs have an increased risk of bleeding with triptans as medicines. Is there any other medication that can be recommended specifically for acute migraines and prevention?
There is a webinar on our YouTube channel. I'm not sure if that's the one you're talking about, but that's specifically about headache. It's Dr. Girortra, Girortra, presented just a few months ago on different methods of treating pain, but especially on treating headache, including migraine. And yes, there are alternatives to triptans. They're more expensive, and some of them are injections rather than pills. You'll want to watch the video in its entirety in order to get that.
Thank you. And so, Connie, I mean, you've been speaking for over an hour, 10 minutes. Thank you so much. We have one more question and then we're coming to the end. The last question. Is there anything on your website for parents who have familial CCM to help them in making decisions about having their children tested? So pros and cons of testing in childhood to help them in making these decisions, as there's no standard guidelines.
Yes. There are several sections in our websites. On our website, there is, in the community and care section, there's a page called Children with CCM, something like that. And then there is an entire section of our website called Genetics that includes information about decision making about should I have genetic testing. But I believe the page that has the specific about children in the care and community section has the more in-depth information about making decisions about testing. But it's always an individual decision. I do want to say, though, that if you are a parent with a CCM3 mutation, your child needs to be tested. That's not negotiable. But with CCM1 and 2, then you have to, it's a conversation.
And building on what Connie said, we also have resources on that on the CAUK website, and the NHS does offer you genetic counselling, because we recognise there's a lot for you to weigh up about whether or not to undertake that testing. And as Connie said, you know, it's a very personal decision. So thank you for asking that question. So I'm just going to double check we haven't had any more. Oh, one I said very last, but is this just linked to the last question, was how early would you test with CCM3?
As early as possible. A cord blood sample, you hadn't thought of that, but you could. But I mean, you can get a blood sample from a baby. So I can share that my daughter had her first hemorrhage at 4 months. I've known other children with CCM3 mutations who had hemorrhage even earlier. So if you can start the testing process right away, you will at least know things. And it becomes important again, because the CCM3 patients' guts are different. And so if you are for example, not breastfeeding using formula, and you're using concentrate formula, you need to be very, very careful about what you choose, because some of the formulas have emulsifiers in them, and so you want to make sure that what you're giving your baby protects their gut lining. And that's for CCM3 children probably more critical than for other kids.
As you say, Connie, it's interesting, isn't it, the importance of the gut. You think we're talking about the brain. Why are we talking about the gut? But as you've talked to, there is a relationship there, and it's really important to be mindful about our gut health.
And then the other, and I'm going to keep talking, Helen, whether you want me to or not. The other thing with CCM3 is understanding what you're seeing when your child is having symptoms. So many of us, our child's first symptom was projectile vomiting, which could be mistaken for many things. If you know they have the mutation, again, they're babies, they can't tell you what's going on. They can't tell you they have a headache. Then at least you can alert your provider to my child has a disease that causes brain hemorrhage early in life. The child is projectile vomiting, do something, as opposed to many of us who wound up having several days of non-intervention because everything else needed to be ruled out.
Connie, thank you so much. And, I'm really appreciative that you gave us that extra time at the end, and your passion shines through. And what is incredible is how you took that lived experience with your daughter, brought it to what was Angioma Alliance, now Alliance to Cure. And then not only that, you make time for people in the UK too. So, we said this earlier, the importance of those international partnerships, because with a rare condition, the only way we're going to move forward and achieve our vision of a cure for Cavernoma is working collaboratively. And I'm just left, as I was last time you presented to us, hopeful. There's so much hope and optimism there. And I think it's been a wonderful gift that you've given us today to expand that and to leave us sort of hopeful for the future.
Well, thank you for having me. It's been a pleasure.
Oh, thank you. So everyone who is on the session, we're going to be bringing this to an end now. We've got one more at 4 o'clock. So I'm going to put up a holding slide, and we're going to be looking at pediatric neurology with Dr. Greg James at 4. Connie, thank you ever so much.